My name is Lekshmi. I'm an environmental science student at Northumbria University, and today we'll be talking about the relationship between honeybees and Himalayan balsam. Controversial topic, the gossip of the conservation sector. So, where do I start? So, this thing about science is the more you dive deep into this one subject, the more and more it makes sense. Like it's like jigsaw pieces falling into place, making this bigger picture. And so this last summer, when I did this study, I got a chance to fall in love with science all over again. So today's talk will have all of these, and I'll start with a little background on Himalayan balsam and honeybees. So, Himalayan balsam, invasive species, also known as policeman's helmet. Well, I had to make that little figure to make sense of the name, and I hope you see it too. So, Himalayan balsam, scientific name, Impatiens glandulifera. It is a large annual plant. It's native to the Himalayas. It is invasive here mainly because it's just not kept under control. So, it outcompetes native plants by growing up to 3 meters tall. So it takes off that light and space away from them, and that way it disrupts ecosystems. And it loves like very damp, wet soil and like readily available source of water. So it loves growing next to riverbanks, and in that process it easily erodes riverbanks. So it's ended up as an invasive species in many areas of the northern hemisphere through human introduction, and that's where I'll introduce this one person called John Forbes Royal. He was a British botanist who was born and raised in Uttar Pradesh, which is a state in India. So he ended up taking charge of this botanical garden in Uttar Pradesh. So he mainly focused on medicinal plants, out of which Himalayan balsam was one of them. So basically collected seeds and shipped them to Britain, Himalayan balsam seeds to Britain, so that the Kew gardens could use them. And it just, from there, one thing led to another. It was used as ornament plants. So it's basically spread out all over the northern hemisphere now, and it's invasive in many. So as you can see, that was the native range. That's the non-native range. So there's been a couple of effective means of control, which has been uprooting and cutting. You would know if you've got in touch with Urban Green. They've been doing a massive amount of work with that Jasmine. And there's also been a research about this fungus, rust fungus, which keeps it under control in Himalayas. So the first thought that I had when I first heard about it was, is it harmful for native species, or does it just keep Himalayan balsam under control? So apparently it's actually not harmful for the native species. It just stunts the growth of Himalayan balsam and keeps it under control. So why is Himalayan balsam so successful? Well, it does produce 800 seeds per plant, which I think is plenty to be invasive. So these seeds, just on this corner, it's very explosive. So in that process, it disperses the seed up to 7 meters away, and this 7 meters away, it's easy for the seed to catch on to a human, or just fall into some water or some animal, which takes it to a new area, and it will easily colonize new patches. And it's also very tolerant to a wide range of habitats, from wetlands to riverbanks and even road edges. So I really don't see any challenges against Himalayan balsam. So as I said before, it also grows quite rapidly, up to 3 meters, taking over everything that the native species needs. So yeah, also lack of natural predators. Well, I guess if you bring rust fungus, I just think like, is it actually a natural predator? But yeah, when it comes to pollination, like pollination is obviously like the biggest part of fertilizing a plant. Like all of its reproduction is just based on pollination. So Himalayan balsam, that little tube that you see of my drawing of Himalayan balsam at the bottom of the plant. Oh, I could probably show on here. Yeah, so just this over here. That's where the nectar collects. So there's plenty of nectar. If you've walked through a patch of Himalayan balsam, you could easily smell how wonderful it smells. And if I was a bee, I would straight go for it. I wouldn't even look at the native plant. So Yeah, so it just wins all the pollinators in Britain at the moment. So that's one of the reasons why it's invasive again. So the star of the show, honeybees. Apis mellifera is the scientific name. Honeybees are social. They live in large colonies made out of one single queen, thousands of female worker bees, and hundreds of male drones. And these bees play a crucial role in pollination and maintaining the health of ecosystems. So we've probably heard all of these things about honeybees, but do you know what the relationship is with Himalayan balsam? They are seen foraging Himalayan balsam a lot, so we're going to find out why. So they produce plenty of nectar and pollen, but so do the native species, native plant species. But the thing is, towards the end of summer and the early fall, that's when the native species nectar creation sort of goes downhill, and that's when Himalayan balsam is at peak. So honeybees go to them as their resource for sustenance and to take back to the hive. So when it comes to the nectar and pollen of Himalayan balsam, honeybees love the nectar, they have it, that's absolutely no problem. But when it comes to pollen, it's just weird, because they don't actively collect the pollen, but because of the structure of the flower, it's like a little tube which is the structure of the flower. Like the bee goes in to get the nectar from the bottom, and then it has to reverse back out because of how small the space is. So when it's coming back out, it brushes on so much of the pollen, like it's thousands and thousands of pollen in one flower anther. So it's literally like, I've touched the Himalayan balsam's anther, and it's like wiping chalk off a board. So all of these bees that collect this pollen on its back, they come out like this. It's like, so they've got this name called ghost bees. So these ghost bees return to the hive after their foraging trip. And these our local beekeepers have seen these bees coming to the hive white, going into the hive, and coming back out all clean. And then we tested the honey samples, and apparently there's hardly any Himalayan balsam pollen in it. So where is this pollen going? It's a bit mysterious. So does it end up in the nectar? Is the bee having it from the nectar? Like, does the bee take it up? Is it ending up in its gut? So we're going to find out where the pollen is going, and we've particularly focused in the study if it's ended up in the gut of the bee. Sorry, I keep saying we, it's mainly Rinke and me. Rinke was my supervisor. So the methods that I used to find out where the pollen's going involved a mix of fieldwork and lab work, and this was Pruda, one of the fieldwork locations. So Pruda was the rural area that we looked at, and Jasmandine was the urban green space that we looked at. In Jasmandine, we went to White Bridge and headquarters. Headquarters is next to the children's park, and in Pruda, we went to Riverside Park for the Himalayan balsam, and Pruda Green for Roseberry Willow herb and creeping thistle, and Whittledean for knapweeds. We just wanted to compare how the bees collected pollen from those flowers, see if they don't have enough pollen in them, like if it's more Himalayan balsam pollen in them compared to the native species. So we focused on catching and collecting bees on sunny days when they're usually out from mid-July to mid-August. And we also, in the fieldwork process, observed the activities of the foraging bees. So what they were doing while they were foraging. Oh, that's a little time lapse. And then came lab work. So we collected a total of 112 bees, and out of this we dissected 64, and those were the honeybees and bumblebees that we collected, just to see if different pollinators act differently towards Himalayan balsam. So the first steps were to identify the species and identify the gender and sort them into little vials, which is shown in this photo, this little tubs. And we froze them as soon as we could after identifying them, because we wanted their organs to be intact before dissection. So after freezing, we took them out again, put them on a dish on melted beeswax just to stabilize, to dissect. So we put them under this dissecting microscope, which zoomed in like 10 times so that we could look up on the organs a bit closer. And we used these sharp sharp tools to cut out this very delicate organ. So we were trying to cut out 4 organs in total, which are mainly the honey crop, large intestine, small intestine, and rectum. So just to give you some background, I'm going to give you a little anatomy tour of the honeybee. So this is the head here, this is the trachea, the chest, and that's the honey crop. I never knew a bee had a honey crop before this project, so that's where the nectar is stored first. And then this little valve, this is esophageal valve, in between the honey crop and the large intestine, called the proventriculus. It is a big word, but just be with me here. So proventriculus, it decides, it's got 4 lobes, and it takes up all the nectar, and it basically processes and sorts and decides what goes into the bee's body, because all of the nectar that the bee collects from the flower, most of it goes back into the hive, and the pollen as well, it goes to making the substance called royal jelly, which the brood or the baby bees have in the nest. So when the sorting is happening, only a portion of what they're getting from the flower goes into the body for their sustenance. So the proventriculus basically decides that, and that if it goes through, it means the bee's got it ingested for its own body. So hopefully everyone's got where the honey crop, large intestine, small intestine, and rectum. So before I continue, I just wanted to show you how every experiment, there's a little trial and error happening. So that's this little thing that we sort of modified in our experiment, which was submersion of the dissection, like before dissecting it. So when we kept it on, stabilized the bee on beeswax, this was before submerging the bee, and this is after submerging the bee. Before submerging, we're using these sharp tools to cut out what we can barely make out there. But here, you can beautifully clearly see the honey crop, this big balloon, the proventriculus, which is just this valve. It's literally just here, this yellowy little substance. And then there's the large intestine going into the small intestine. And it's so clear, and it was so much easier for us to harvest them. So, in case nobody has come across a bee dissection before, you might not see it again. So this is raw and original. I took this photo. So yeah, this is the honey crop. That 2D image is basically a 3D here. So the honey crop, the proventriculus, all of these organs just working together to keep the bee alive. The Malpighian tubules, just to add on to that, is just a structure that comes out at the junction where the large intestine and small intestine meets, which acts as a kidney for the bee. So the thing is, we took these photos to keep track of what the honey crop's looking like when we dissected them, because when we look at the honey crop of a dissected bee, we get to know what stage of foraging they are in. So this one's probably like, they've left the hive, they've done some foraging, they're not going back home yet. But this one, look at the size of that. It's definitely going home. It's gotta go home. So we've got this bumblebee, which we collected from Himalayan Balsam in Jasmine, and this one's a honeybee from Jasmine. So we can also see like there's a structural difference where the small intestine is quite longer for the bumblebee as well compared to this honeybee. So we basically dissected 4 of these organs, put them into little vials, again froze them, and put them onto slides to look at them under the light microscope. The light microscope, sorry. So there was a lot more magnification into this, and the first one's basically a close-up of the proventriculus, which is the valve that I told you about, and the 4 lobes, and this is like clusters of pollen inside. So it shows that the bee's got all of this pollen inside, like being processed inside the proventriculus. And when we magnified into that photo, it basically showed a couple of Himalayan balsam pollen, so it probably does take it in. So this is a honeybee. All of these bees were collected on the same day from Jasmine. So this is the honeybee's proventriculus, and this is the same honeybee's large intestine. So you see this pollen here. This one is rectangular with 4 pores, 4 little gaps in the corners. So that is the Himalayan balsam pollen. So when we look at this one here, we can see an abundant amount of rectangular and oval looking pollen, and most of it is Himalayan balsam. So what does that show? It shows that it is going into the body of the bee. So we were before we were asking ourselves like, oh, where is the pollen going? So now we know it's passing the proventriculus from the honey crop and actually going into the bee's gut. Compared to the bumblebee here though, this is the bumblebee's large intestine. Same day, same crop, same organ, large intestine, same patch of Himalayan balsam. And this one's a bit more round, it's a bit more flappy, the pollen, so it's definitely another family of flowers. So this was from a carder bee that we found in Jasmandine. This was on the hind leg. This is basically a pollen basket close up. If you don't know what a pollen basket is, it's basically this chunk of pollen that the bee, when it collects when it brushes off the back or collects from the flower, it sweeps from the front legs to the middle, to the back, the hind leg, and it sticks it on its thigh to take it back to its hive so that it can feed it to the young. So the thing about this pollen basket here is that it's very pale and quite white almost, which shows that it's been taken Himalayan balsam pollen, which is very prominently white. So the findings of this project is that we observed 3 bees actively collecting Himalayan balsam pollen, and we saw most of them evading the anthers, which is by just escaping through the side of the petal. They didn't even bother looking at the anthers, like they just didn't want it. And then we also spotted a lot of ghost bees, which are the bees covered in white, and we just found out that honeybees do ingest the pollen and it does end up in the gut, but we don't know if it's because of the structure. Like it is a bit droopy and the anther comes at the top, the nectar comes at the bottom, if it falls in, and if the bees just drink in the pollen with the nectar. So to conclude, our research revealed a diverse array of behaviors of honeybees interacting with Himalayan balsam, but they do ingest the pollen along with the nectar. So the next step in the project, because I've taken this internship as a dissertation and I'm starting data collection again this summer, we're going to be looking at new honey samples from local hives near Himalayan balsam patches and find out why honeybees might like Himalayan balsam through nutrient analysis. So we would be able to find out why they might like it, like the protein content, what nutrients they prefer, I'll have to study that. Anyway, I'd also like to quantify the importance of Himalayan balsam pollen for honeybees. So that's by counting the particular types of pollen in like each of the slides, that like the photos that I'll be taking, slide photos, and I'll be looking at the number of each type of pollen per organ. So thank you so much, One Planet, for this project and funding the project, NERC for actually giving me the project, and my 2 coolest supervisors, Rinke and Matthew. This project wouldn't have been this amazing if it wasn't you both making it this amazing. So these are just the references that I've used for making this presentation, and thank you very much everyone for listening. I'm open for questions now.